We've been talking about gag molecules, and so far we have basically talked about them in context of structural entities. Let me say this emphatically, that they are not there just for structural support. These molecules have a very important role in physiology, and let's look at some of the examples of some of the roles these molecules play. So first of all, as we have discussed, they form gels of varying pore size and charge densities. So these molecules, or the gels made by these molecules, they can serve as selective sieves. So just as you use a sieve to filter, for example, when you're pouring tea, from filtering out the tea leaves from the fluid molecule, so these sieves in a similar fashion can select what molecule can pass through and what molecules cannot enter. For example, of course, if they're negatively charged, they will resist negatively charged entities from going through these gels. So one of the things these molecules do is they regulate traffic according to, of course, their size and charge. Heparan sulfate proteoglycan called perlecan in basal lamina of kidney glomerulus helps filter molecules passing from bloodstream into the urine. So here's a specific example of these molecules acting as selective sieves. Proteoglycans are thought to have a major role in chemical signaling. We have discussed cell signaling in great detail, as you will learn from this module also and other future modules, that these molecules also have a very significant role in cell signaling. For example, proteoglycans can bind various secreted signal molecules and growth factors, and either this binding can enhance the signaling ability of these ligands, these signaling molecules, or they can inhibit the activity of these signaling molecules. We'll look at some very specific examples in a later module, but I just wanted to explain some of these things in this module also. For example, heparan sulfate chain of proteoglycans bind to fibroblast growth factor. We have talked about FGF. It is one of the signals that binds to receptor tyrosine kinases, for example, and it can stimulate various cells to proliferate, meaning they could divide. Oligomerization of growth factor molecules with the help of these proteoglycans, for example, helps. For one thing they do is they can act as a reservoir. They can hold these signaling molecules in appropriate locations. As you can see here, this particular gag is holding the signaling molecules. Also, since this core protein, which is a transmembrane protein, it will have more than one gag chains, and these gag chains can attach to FGF at more than one locations. This also helps the receptors to come close together. So they basically enable growth factors to come together, these receptors to come together, and enable them to cross-phosphorylate each other. We have talked about this cross-phosphorylation and how it serves as a dock for other additional proteins that can relay the signal to the cellular nucleus. So basically, this is one of the roles of these gag molecules. Through gag molecules or proteoglycans can basically help cellular signaling. Some members of transforming growth factor beta or TGF beta, they bind to the core proteins of several matrix proteoglycans, including decorin. We have talked about decorin as a protein, small protein, which has a single gag, and it binds or basically decorates collagen fibrils. And because of this binding, decorin inhibits the activity of this particular growth factor or TGF beta. Decorin also has a role in regulation of autophagy of endothelial cells and inhibition of angiogenesis. Inhibition of angiogenesis means angiogenesis is a process which basically results in formation of blood vessels. So decorin, which is a proteoglycan, it is regulating autophagy, which means cells eating their own parts in endothelial cells, and also it inhibits formation of blood vessels. Heparan sulfate secretes chemotactic heparan sulfate, which is present, for example, on surface of endothelial cells, It immobilizes secreted chemotactic attractants, which are molecules which will attract a specific cells on the endothelial surface of the blood vessel at a site where inflammation or injury has taken place. It will hold these molecules there long enough so white blood cells can come and recognize this part, and they can recognize that there has been an inflation or they are needed there. So it is basically acting as a pole on which a flag will be mounted. So and if signaling molecules don't stick to these proteoglycans, they will just be washed away in the bloodstream. That doesn't happen because these molecules can grab these particular chemotactants for white blood cells and hold them at the site of inflammation, and ultimately the white blood cells will come there and they will do their job. So binding, so we are on the subject of proteoglycans, and we are talking about their activities more other than the scaffolding function. So let's see these 5 different ways and they can perform other functions. As I have talked about, immobilizing proteins close to the site where it is produced, thereby restricting its range of action. So if a protein signaling molecule has been produced in a location and cells want to hold it for some time, they can act as immobilizer of signaling molecules. They can sterically block the activity of a protein. So if an enzyme, for example, is glycosylated near the active site, it can basically inhibit the activity of that particular protein. They can provide a reservoir for a delayed response. So these signaling molecules or specific molecules can be absorbed into proteoglycan structures, and the body can hold them there till they are needed, and at that time they'll be released from this reservoir. They can protect proteins from proteolytic degradation, thereby prolonging their action. For example, we talked about protein enzymes and lysosome that can degrade proteins. Logically, it would make sense that if we have proteases, bunch of proteases sitting next to each other, why won't they just eat themselves up? One of the reasons is that they don't eat themselves up is because these proteins are glycosylated. Remember, they came through the ER route. So these sugar molecules are protecting the protein cores from proteolytic degradation, especially for lysosomal cells. Concentrating proteins for more effective presentation to the cell receptors. We have seen an example of, for example, FGF, that how these molecules can grab FGF and they can present these FGF molecules to the receptors and allowing the receptors to cluster and produce a more robust response. So we will continue our discussion of proteoglycans and GAGs in future modules.